Hi, everybody. It's really been great to hear, the morning's presentations and see how far that FaceBase has come and the plans for all the great things that'll be happening through this project into the future. So our project includes myself, Elizabeth Leslie, who's here a few rows back, Elizabeth, also Eleanor Feingold and Harry Hockheiser, who could not be here today. So this project was motivated by a number of sort of converging levels of need for this project. So in the first phase of FaceBase, there were 3 projects that included developing genomics resources, a couple GWASs and another project that had GWAS and some sequencing. So as we were thinking of ways to best display the results from the parts of those initial projects, we started thinking about something along the lines of what this project turned into. Another reason that we really were trying to think of ways to have a tool like this is that all of us involved in human genomic studies get many emails from people saying, I have this SNP I'm really interested in, or this gene I'm really interested in. Could you just look it up real quick in your data and tell me what it shows? What kind of p-value did you have? What do my results mean? And our group and our colleagues have lots of large sets of data like this. People who come to us with those questions could just go to dbGaP and go through the process of getting access to our data. Most of our data is on dbGaP, and they could get the data sets themselves, the original raw data, go through the process, which takes quite a few steps. So it makes it kind of a barrier to people who would like to do that. You have to demonstrate that you have a secure data system, that your RT people have been trained in the particular security processes that NIH looks for. You have to have, in some situations, IRB approvals to be able to then analyze that data when you get it. And you have to have a team of analysts who know how to analyze that raw data to allow you to look at the result for that one SNP you might be interested in. So that's, again, a barrier for a lot of people who are interested in knowing what the results are from our genomic studies. Then some of the big genomic databases out there are from other countries, so they're not even on dbGaP. So we really started to think about how we could do this in a way that would be very user-friendly and allow people to do lookups. And honestly, now that we have this tool, we're one of the big users. We're always thinking, well, what did it show with that gene? So we'll go back and to look at our own data using the tool. So the goals of the project, again, were to devise a web-based interface for looking up results from human genomic studies without having to go through the process of getting access to the individual level data that is available through a very secure hardware interface. Then once we develop this tool, we then need to load it up with results from craniofacial and oral genomics projects that are of interest to the FaceBase group and other people involved in craniofacial research. And we've identified these by working with the research community, looking at dbGaP, looking at publications as they come out. Then we came up with some strategies to create and display results from these genomics projects that are meaningful to most people who are looking for these results, but not so complicated that it's hard for the users to grasp what we're depicting. And the 2 major things that we are using in that regard are Manhattan plots and results from locus zoom, which are basically zoomed in Manhattan plots. So our approach to keep things, okay, closer now, sorry. So our approach is an attempt to be as user-friendly as possible to save user time and effort, to pre-calculate results databases. So that was a bit of a change from when we first envisioned the product. At first, we thought it would be the most flexible to be able to calculate results on the fly. But as we started looking into it and thinking about it more, there's an awful lot of ways to go wrong in setting up analysis of SNP data. So we decided that at least for the first bolus of data sets that we've uploaded into this tool, we would pre-calculate results databases. For example, p-values at all the SNPs that were run for that particular project. And that means calculating a lot of different results databases. So databases for relevant subsets. So for example, many of the projects in cleft lip and palate have multiple ethnicities. Somebody might only be interested in the Asian results. They also often have both genotype SNPs versus imputed SNPs. So we have subsets that have just the genotyped versus all the genotype plus imputed. You might have specific sub-phenotypes you're interested in, and in some cases we have environmental data that we also did subsetted results databases for. Prepare Manhattan plot files from these results databases. Then for the specific results for SNP genes or genetic region requests, we do have that set up to be done on the fly in a sense. I mean, it still probes the pre-existing p-value data sets, but we did not pre-calculate locus zooms for every single location of the genome. You enter what you're interested in, and it gives you back the result. So to date, we established the hardware and the computer system early in the first year and set up the basic web-based software. We've created a website, and that's been updated periodically, and multiple projects have been uploaded in oral facial clefting, normal facial variation, and a few oral and dental data sets. We have project pages, result databases, etc., which I will show you examples of. Now, one thing that we're in the middle of doing is improving our locus zoom functionality. So the way we had implemented it in the website was very slow in execution. So there's now a local version of locus zoom, which we'll be planning to replace our current locus zoom with. So let me apologize to the audience. Anybody who's trying to use our site for the next month or so will not really be able to use the locus zoom functionality. So sorry about that, but it'll be much better when it comes back. In the process of replacing the locus zoom, one reason it hasn't happened yet is that midway in that process, we lost 2 programmers, kind of a programmer and a half in terms of FTEs. So we weren't able to immediately replace it as we were hoping to. Then we also are at a stage where we have several results databases that are just ready and waiting to be uploaded from a number of projects that are quite recent. So the PIs on those projects want the papers to come out before we upload the data for public viewing and analysis. So we have some new clefting GWASs from China and African GWAS. We have a number of new US GWASs, or more specifically, different ways of utilizing the GWASs that exist. There's a couple different facial variation GWASs that build on the FaceBase facial variation GWAS. We have an earlobe morphology GWAS coming out, and some Scandinavian dental GWASs that we'll be able to upload. So we didn't add much new in terms of databases this year, but we got a lot of these ready, and hopefully very early in the coming year, we'll be adding those to the tool. So this is the direct link to the website with the tool. There's also links on the home page of FaceBase and on the FaceBase project page for this tool. So this is what you see is the landing page for the website. So our logo, then it has the 3 basic areas that we have projects available through the website: oral facial clefting, facial variation, dental traits. It lists it. Would take you to FaceBase if you click on the FaceBase, and gives a little bit of the information about the motivation of the project. So if you then were to click on the oral facial clefting icon, you would see a list of the projects that are available through the tool. So this particular screen shows 4 projects, and like I said, we have additional projects in the works. Spatial variation, we have 2 right now, but multiple additional projects in the works. The dental traits, 3 right now, and have, again, some additional ones that are coming up soon. Okay, so now if you are interested in a specific project, so now we're back at the oral facial clefting project page, and let's just click this international one, in part because that was involved in FaceBase 1. So you first see, actually, sorry, this is from a different project, but still it's an oral facial cleft project. So there's information about the project in general, so a description of it, plus the funding that led into the project. There's a methods tab, so when you look at the methods tab for this one, it sends you to a brief page where there's additional links for finding the methods that were used for the data displayed here. There's a Descriptive Statistics tab, and each project has different types of statistics that are of interest. This particular project has several of the various study variables that are available. One that's of particular interest is how many individuals in the database have cleft lip alone, cleft lip with cleft palate, cleft palate alone, etc. So if you want to scan to see what type of variables are available in the data set, that's shown here in the descriptive statistics. Then this is the real meat of the tool. And this is what we call explore the project data. So every project is a little bit different, but they have some things in common. So it's first you can pick which set of the data you want to explore. So for this one, we had the genotyped GWAS panel, the imputed panel, which includes the imputed SNPs. There's multiple ancestry groups. You can look at all of them together, or you can look at individual ancestries. In the phenotypes, for this one, you could look at cleft palate alone or cleft lip with or without cleft palate. There's a couple of different types of relatives or families in this data set. There's a set of trios, and then there's a set of singleton cases and controls. So you could look at the analysis results just in the trios, just in the case controls, or as a meta-analysis, including both the trio and the case control data. Then you can say if you want to see the Manhattan plot, or if you want some more specific results. This other project, Genes and Interaction, Controlling Oral Clefts, you can see it has a little bit different interface for the things you can choose. Again, genotyped imputed. This was genotyped back on build HG18, but it was also translated to HG19, so you can look at either one. Again, different subsets of the phenotype, different ancestry groups. This one is unique that it has the environmental variables, so you can subset your GWAS results by whether the parent smoked, whether the mother smoked during pregnancy, used alcohol or vitamins. This one shows results from some targeted sequencing in particular regions in clefting families that are either Asian or European. And again, you kind of get the idea that these are really tailored to what the project did, and to some extent what the PI is willing to have on display. If they've got paper out, but they've still got more paper coming, they might be okay with us showing the results from the paper that's currently out, but want us to wait for the upcoming papers. Again, same thing, just kind of tailored to the project. So obviously this is a Manhattan plot coming out. So this comes out as a PDF, which you can download if you want a PDF to include in a paper or a poster or something. This is one just using the imputed data, and the titles are meant to be informative about what is being depicted in the particular Manhattan plot. So we also put in some checks. So if someone chose a subset of the data, in this case, the genotyped data from Asians with cleft palate and trios only, and they asked for a Manhattan plot. So this choice of subset has a very low number of trios, so the Manhattan plot was not generated. It was not very meaningful, too low power to show meaningful results in a Manhattan plot. So that's the Manhattan plot side of things. On the more specific way of exploring the data, we allow looking up a particular gene, a particular SNP, or a chromosomal region. So if we put in a particular gene, IRF6, you get a typical locus-zoom plot, again with a title that shows you just exactly what you requested. If you request a region, you have to give the base pair to the chromosome and the base pair to base pair position that you want to be depicted. Shows a little more clearly. And again, the locus-zoom will come up. Okay. So again, the initial project description includes some information and includes an active link to dbGaP. So when you click that, it just takes you to the dbGaP page for that particular project. And that also provides an easy way to go to exactly the dbGaP project if you do want to end up downloading the individual level data. That's another purpose that we saw for this tool. People might use it to do some initial scans through data to see if it has the degree of data that you're interested in to make it worthwhile going through the process of downloading the individual level data from dbGaP. Okay, and the final tab is publication. So we have an active link to PubMed that will search for all publications that are associated with the particular project. We do that by the grant numbers that funded the projects. Okay, so just a little update about traffic to the site compared to, so in blue, we have the period from May 1, 2016 to February 2017, compared to, in blue, compared to the previous year in orange. So you can see we've had kind of similar, so if you look at just the number of sessions that were started in the two time periods, it's really similar. There was a slight uptick in the current year's amount, so almost 2% more people visiting. The number of individual users, though, was increased by 18% from the period last year, and we have about 840 unique users, and that's subtracting out the people from PIT who are, go on it for development and so on. Then it's kind of interesting from the current time period, there's a higher proportion of new users than there was in the previous period. And our bounce rate has improved. It's less than it was last year. It was like 78%, and now it's down to 44%. So that's good to see. And maybe that bounce rate is improved because the number of people from Russia visiting the site has gone down by about 50%. But the distribution, so people visit the site from around the world, mostly the US, but also UK is a heavier user and really spread out all over. So in the future, again, we'll be updating the locus and trying to use it right now while it's down. We have these data sets that we'll be adding. We're working on ways to be sure people cite the project in their papers, and would also like to add to the dbGaP project pages a link to the tool so that people might find a data set they're interested in dbGaP, and if it's represented in the tool, they would be able to scan the results databases that way. Then there was some additional functionality that we had talked about when we were initially coming up with the tool, and that actually came up at last year's meeting, because something that users would like to see that we do not yet have is ways to look at results from multiple data sets simultaneously. So that's a challenge for the coming year that we hope we can come up with some additional web design that would make that possible. So in addition to the faculty involved, we also have a variety of staff and students who have been very instrumental to the success of the project. And Tamara Mitchell is the new programmer we've hired, who hopefully will allow us to get back on track with the Locusum stuff, for example. So thanks very much, and thanks to NIDCR for letting us work on this tool and continuing great progress that's happening in FaceBase.
I've got a question for me.
Nobody wants to say, I've got this SNP.